Gliding motility is the type of movement used by bacteria that do not have flagella. Usually they have different types of structures or secreted fluids that allow them to drag themselves along. Gliding motility is typically more slow and smooth than the use of flagella, but it requires that a solid surface be in contact with the cells. Bacteria that do this type of movement are usually filamentous or rod-shaped. There are 4 mechanisms for gliding motility that we will be covering.
The first mechanism for gliding motility that I will be discussing involves the use of slime excretion. Cyanobacteria glide by secreting a polysaccharide slime from pores onto the outer surface of the cell, as shown here. The slime, drawn in blue markers, touches both the cell surface and the solid surface against which the cell moves. As the excreted slime sticks onto the surface, the cell is then dragged and pulled along. Cyanobacteria is actually not the only organism that glides using this mechanism. The non-phototrophic gliding bacterium, Cytophaga, also moves at the expense of slime excretion, rotating along its long axis.
Another mechanism of gliding motility is the use of type 4 pili. Microbes such as Myxococcus xanthus extend their pili and adhere to a part of the surface that is far in front of them. Then they contract their pili and drag the cell body to that point. They can again extend their pili to come in contact with the surface farther in front of them and drag their body to that point again. Myxococcus xanthus have actually been shown to have chemotaxis-like functions for their pili, and so they can direct their movement either away from repellents or towards attractants.
The third mechanism of bacterial gliding is to use a protein adhesion complex. The protein adhesion complex will form at one pole of the cell, especially of the rod-shaped cell. When moving, the protein adhesion complex will move in the opposite direction of the cell, while the cell glides forward with the help of the friction created by the adhesion complex. Then, adhesion complex will be pulled back to the back pole of the cell. This mechanism is fueled by cytoplasmic motility engine.
As you can see, motility of bacteria are very versatile. I am going to discuss the mechanism of gliding motility of Flavobacterium. Flavobacterium is classified as gram-negative and can be found in soil and fresh water in a variety of environments. Its cell surface has proteins that move to create motility. The proteins are anchored to the cytoplasmic and outer membranes. The proteins move and push the cell around the surface. Protein movement gets energy from proton motive force. When protons move into the cell, it creates energy that causes the protein to anchor and move bacterium the opposite direction of that protein.
Gliding motility allows bacteria to move towards more favorable environments and away from less favorable environments. Although they can't identify specific chemical compounds like movement by flagella can, they can still move and explore more of the petri dish and happen upon more favorable conditions. For example, a colony growing in a compact area of the petri dish will quickly run out of nutrients as they are all consumed. A bacteria that cannot move will run out of nutrients and not be able to move to areas of the petri dish with more nutrients. A bacteria that can do gliding motility can move away from the rest of the colony and find more nutrients that have not been consumed yet. This allows them to survive longer and duplicate more. Another benefit to gliding motility is the ability for bacteria to come in contact with each other more by being able to move. Coming in contact with other bacteria of different species, perhaps in the same environment or in the same petri dish, allows them to do conjugation more. A pili is only so long, and so conjugation may not work if the bacteria are too far apart. However, if one of the bacteria is able to move towards the other one, the pili can come in contact with the other bacteria and a plasmid can be transferred, and the bacteria may gain more genes that it did not previously have access to.